Super. Okay. So this is going to be our weekly review of the slides and the lecture materials and all of the things that we did. We did a lot of active stuff, and we just want to make sure that you're able to kind of take the activities that we did and apply it to some of the main concepts that we're exploring in this particular unit. So our learning outcomes were really sort of starting to ramp up a little bit, related to being able to recognize which variables, which factors are important in a scenario and a story, to understand how the biological levels of organization can be applied to some of them. And so we didn't talk about that specifically. What we want you to do now, though, is to take that learning outcome and start to apply it yourself and figure out how it matters. And then we also did some data collection, and we did a whole bunch of things. So it probably feels like there's a lot going on, but we'll do a very short summary of everything. The first thing that we did was we threw a whole bunch of terms at you, and we asked you to organize them. And some of you did that, which is great. We've been looking at some of your flow charts. There are a couple of things that we would like you to recognize here. For example, there are some sort of more general terms, random and not random, that we would like you to use in that flow chart. And the other thing is that there are two gene flows. So just notice those things. They're done deliberately as you try to organize them. Now, I did very quickly flash my organization here, and we're not going to focus on it too much because, again, we want you to do this and get it right in your head. We will post it right before the midterm, but it's way more important that you go through this process yourself in order to come up with it. But it should look, or could look, something like that. Then we went through a bunch of sort of baseline facts, things that we think are really fundamental to understanding some of the concepts and some of the things that you can use to distinguish the concepts. Questions about where variation comes from, whether they're random or not random. Mm-hmm. One of the things that I kind of struggle with, especially when we think about all of this, all of the different terms and everything, it says here variation can change within populations in various ways. And for me anyway, I don't think we've ever talked about this, but for me, mutation kind of stands out as being something that's somewhat different than the rest of these. Because in my head anyway, and I see how variation can change with the rest of these, but ultimately that variation comes from mutation.
Indeed.
So for me anyway, it kind of sets it apart. Does that make sense?
And it also is, when we talk about natural selection, we're going to talk about how it sort of has two steps involved. The first one is getting the variation in the first place, which is from mutation usually, and then the process of what happens to that variation after it arises. So they are different in the sense that mutation can be a prerequisite for the other ones, or it is fundamentally at some level a prerequisite, because you don't have variation without mutation. The only thing that is potentially kind of similar is that gene flow can add variation to a population, and so can mutation. Natural selection, as we've been defining it, a certain kind of natural selection, and genetic drift both reduce variation in a population. So there's ways you can pair them up. That might be actually a good activity, is which ones have similarities and differences, and why might one stand out or another. But fundamentally, those are the four mechanisms that can change the nature of variation in a population. Mutation is a little bit different because it creates new variation. The other ones are more about reassortments of existing variation. So yeah. I agree, that's a little bit different.
Cool, yeah. So actually, if you take a look at my my flow chart, mutation is up here. Because for me anyway, that's kind of a fundamental requirement, but it may not be for you depending and that kind of deal. So
depends on the question. So there's that theme of you'll run into all the time in this course, which it depends. So for example, if I have a scenario where I have a rare allele that persists in a population, mutation might not have any more explanatory value in that case than genes moving in from another population. But on the broad scale of evolutionary mechanisms, it is sort of different because it makes the others possible.
Let's check to see if it's still recording.
Let's do that.
Yes, good.
Yeah,
sometimes it cuts out, so okay.
I will cut that part.
Moving on.
So, mutation.
So we went through a bunch of different types of mutation, and actually the most valuable contribution that was made to this slide was that one of you came up after class and said, well, what about insertions or deletions or all of these other types of changing of the genetic code? And you're totally right. This is not a comprehensive list in any way. The point of this list wasn't for you to know every single one. The point of this list was to show you that certain types have different implications at different levels of biological organization. So that's way more important, way more valuable, I think, from this slide.
Yes, and just to clarify, I guess, on that point, insertions and deletions occur at a variety of scales and by different mechanisms, but those mechanisms are point mutation, chromosome mutation, gene duplication, so unequal crossing over, unequal sister chromatid exchange. There's lots of reasons that you can get deletions and insertions, but they fall into those categories.
Cool. And then this is really the only time that we formally talked about natural selection, because I think we agree that generally, if anybody understands anything about evolution, it's going to be more about natural selection than anything
the very,
else.
very basic notion of it.
Yeah.
We'll see.
Yeah. So it's funny when you talk with people and you say natural selection, they say it's survival of the fittest, and when you ask them the fittest what, they tend to say species, which is a subtle problem.
Probably one of the top 10 misconceptions about natural selection, but anyway.
That's right.
We'll get to all that. All that will be made clear in time.
Yeah, we'll get to that. But anyway, so we haven't spent much time on natural selection yet. Everything that we've talked about from here on has been on the random side of things, the random side of evolution. And so we focused on genetic drift. Genetic drift being an umbrella term that describes the random change, or for random mechanisms, the random change in allele frequencies. Understand that the two examples that we then explore, the bottleneck and the founder effect, I'll just go through these very quickly. There we go. The founder effect and the bottleneck are examples where genetic drift can happen to a very extreme end. Genetic drift can happen to a non-extreme end as well, but we're going to be exploring those two mechanisms more specifically, okay?
Genetic drift, just so you know this, it's not part of the scope of this course, but you'll see if you carry on. Genetic drift is always happening, and the relative strength of genetic drift versus other processes has a lot to do with population size. So you'll learn more about that as you go along. So genetic drift is kind of always happening. The extreme examples that we want to point out are founder effects and bottlenecks, because they really illustrate the random side. I would point out you should make sure you're pretty clear on what random and non-random mean in this context. There's important ways of understanding those terms, so that might be something to put on the discussion forum, for example.
And then we left off with this table, and we asked you to try to fill it out. And so far you guys are doing well from the tables that we've seen.
Mm-hmm.
Some of you, though, are really able to demonstrate some misconceptions, which is really nice. So, for example, the whole idea about whether or not natural selection increases or decreases variation. Mm-hmm. The answer to anything in biology is it depends. But generally it is one way. And so some of you are struggling with that, and we see that as a really positive sign.
Yes, and the two things I would, I guess, say about this table, so one of them is natural selection as we've been talking about it, which is a form of natural selection called directional selection. We won't get into too much detail about that, but it's where you're losing certain individuals or others are out-reproducing some individuals because of specific traits. That leads to a reduction in variation. There are other manifestations of selection that can maintain variation, but we're not talking about those, so you should be thinking about reductions in variation at this point. The other thing is, I'm going to go back just for a sec to the table. We've had a couple of questions about the two columns on genetic variation and genetic differences. The first one, the second column here says genetic variation. That's referring to what's happening within one population. Genetic differences among populations means how different are the two populations from each other. Maybe on average, or in terms of however you want to measure that, but are the two populations similar to each other or different? And the first section there, genetic variation, refers to how many different kinds of things are there in a population, one population.
And if you're wondering and you want an example, you can just take a look at the homework help video for this particular table where we go through and fill out the first one for
Yeah.
you. So just to
we
get
just,
you started.
we just had a number of questions, so you should
Yeah.
check that video out too.
Okay, cool. So these are the learning outcomes from Wednesday's class, and here we're really focusing on how to collect data and what the implications are of your collection methods, and then how to represent data and what the implications are of the representation. And so we went through the homework question, and there's a video that talks a little bit about setting up the scenario for the implications of Veliger versus Glaucidia larvae, so you can take a look at that as well. And to me, it was pretty good because it was a split between the two most likely answers, and then really I think it was just a matter of whether or not you fully understood the implications of gene flow versus versus extinction and speciation and those types of things.
Yeah.
So I was quite happy with the way
Yeah,
that
no,
split
that
was.
good,
Yeah. And then we talked about variation in a population, and I think for me, maybe for you it's something else. For me, the most important thing to explore was this phenomenon of a normal distribution. Why is there a normal distribution in biology, in nature, in everything, pretty much?
Very common.
And what does that mean? What are the advantages, disadvantages? Are there advantages and disadvantages? When does it work? When is it not a good idea to have a normal distribution? These are pretty high-level questions, and we feel like if you can grapple with those, not answer them, but grapple with them, then you're definitely on the right path to understanding the implications of it. And then we played around with some graphing data, which was fun. Thank you for participating in that. That was great. And we were able to generate these graphs and then discuss the normal distribution and when there really is a normal distribution, but the way that you graph it isn't showing it, and being able to recognize when that happens versus when there really isn't a normal distribution, that kind of deal. And our advice, if you want to take this further to make sure that you fully understand it for the midterm, is to go off and find data, find graphs, and come up with true and false statements associated with each of them. Test yourselves, try to trick your friends into getting the wrong answer or getting the right answer, and really try to test each other based on some graphs that you find, and in any subject, any topic will work. So then here are some questions that you can reflect on, both for this exercise as well as for the graphs that you find. Totally fine, just some guiding, studying questions. And then we want you to see if you can relate it back to some of the stuff that we're doing in the inquiry case. So, we've shown you some data with the mussels, we've talked about genetic drift, gene flow, evolution, we've got a scenario where there's an invasive species coming in, so really try to wrap around those terms around the mussel scenario. And one thing that you might want to do is to kind of map out the story of the mussels, what's going on, and then to superimpose those terms either sort of in your mind or on a whiteboard or something, so that you can understand how they're all connected.
And you can think of it this way: the reason we chose this as a good case study for this unit is because it gives examples of all of those different mechanisms that we want to get across. That's why it's one of the things we chose to talk about, because it's very good at illustrating those things. So you should be able to map on the concepts to the story itself. That would be a useful exercise for sure.
Cool.
I like it. And that's it.
That's it. Great.